Hello, everyone, and welcome to today's session of Bite Size Science, how a genetic diagnosis changed Michael's life. For those of you who don't know me, my name is Mara Jean Tilley, and I have the great privilege of heading up the Garvan Research Foundation, the marketing and fundraising arm of the Garvan Institute of Medical Research. It's wonderful to see so many of our loyal donors, partners for the future, volunteers, and board directors in the Zoom room today. Thank you for being with us. We also have a very special visitor today, as I believe Michael, who is the subject of today's talk, is also with us in the Zoom room. Michael, a sincere thank you to you for allowing us to share your very personal story with our audience today. It's a wonderful example of the importance of medical research in the improvement of human health, and we're indebted to you for allowing us to talk about your diagnostic odyssey with our special guests. Thank you. So today we're going to hear about Michael, a 52-year-old man who has suffered or had suffered from a mysterious, unexplained immune disorder his entire life. Michael had lost hope that his symptoms would be properly diagnosed before he came to Garvan and the Circa program. Through the whole genome sequencing and analysis undertaken by our scientists through the Circa program here at Garvan, he received a life-changing diagnosis and, importantly, targeted treatment after more than 4 decades of not knowing what was going on. For those of you who are not aware, Circa stands for the Clinical Immunogenomics Research Consortia of Australia. This is a national, indeed a global initiative founded at Garvan. Very exciting. Sadly, there are many people just like Michael undergoing long, tough diagnostic odysseys for rare and difficult to treat medical conditions, including immunological and autoimmune disease. Understanding the complexities and variants of the genomes in patients with primary immunodeficiency diseases like Michael also helps researchers understand more about how to harness the immune system to fight diseases such as cancer. Before I introduce Dr. Pei Dai to tell us more about Michael's case, it's very important for me to take a moment to acknowledge the Gadigal people of the Eora Nation, the traditional owners and custodians of the land on which Garvan and St Vincent's Hospital are located, and pay my respects to elders past, present and emerging, and a particular acknowledgement that this week is NAIDOC Week. NAIDOC Week celebrates the history, culture and achievements of Aboriginal and Torres Strait Islander peoples and recognises our First Nations people and the fact that they have occupied this land, this continent, for more than 65,000 years. This year's theme, as I'm sure you'll already know, is always was, always will be. A little housekeeping. As you know, the Bite Size Seminar Series is designed to fit into your lunch break or to be a brief Zoom fatigue-free dive into the latest in scientific and clinical endeavour. So we do only have 30 minutes this morning. Accordingly, we're going to hear from Dr. Pei Dai before going into a poll for voting on what questions you'd most like to hear the answers to today. But if you do have any particular questions that are not addressed through the talk, I would encourage you to enter them into the survey at the conclusion of today's session, along with your email address and name, and the team will be sure to come back to you over the coming days as soon as they possibly can. So our special guest for today, Dr. Pei Dai. Dr. Dai is a medical doctor in the final year of his clinical immunology training and a PhD candidate studying with Professor Trifan, the Garvan Institute of Medical Research. Dr. Dai's clinical practice has spanned both St Vincent's Hospital and also our friends at Westmead Hospital, who we work very, very closely with. Dr. Pei is the inaugural recipient of the David Cooper Fellowship and also a recipient of the Royal College of Pathologists Postgraduate Research Fellowship Award. His research currently focuses on studying the genomes of patients with rare, undiagnosed disorders of the immune system with the goal of providing better treatments for patients and increasing our understanding of the immune system in health and disease. Dr. Day, welcome. Thank you so
Thank
much for
you.
taking some time out from your patients and from the lab to speak with us. Now, it looks like you might be in the wet laboratory as we speak.
Mm-hmm.
Fan-
I'm doing lab work today.
Fantastic. Now, before we kick off, perhaps you could tell us a little bit about what inspired you, having undertaken your extensive medical training, to do a PhD at the Garvan Institute with Dr. Tri Phan.
Yes, thank you. So I actually became interested in this area which intersects genetics and immunology after working with Professor Phan at St. Vincent's as a junior registrar and basically seeing and treating patients like Michael. What we found was that these patients often had seen many doctors over many years, and often they've been put into the too hard basket. On the other hand, genetics is it's a relatively young field. What we found was that if we apply what we learned in genetics to diseases of the immune system, we were able to provide diagnosis for patients like Michael at a genetic and molecular level. And it allows them to get closure, but also allows them to get much better treatment from drugs that you didn't think were relevant before. And it also allows us to significantly improve our understanding of the immune system. So overall, I actually found this area very, very satisfying professionally. And that's why I decided, to do a PhD in this area.
It really is a new age, isn't it, of medical research coming together with clinicians to
Yeah.
fast track treatments into the clinic.
Yeah, it is. It's kind of like Michael's case is a good example of bench to bedside medicine where you use laboratory research and you translate that directly to better patient care.
Thanks, Pei. Well, we might let you take over with your presentation. I'll come back to you for Q&A shortly. Thank you
Sure,
very much.
thank you. Okay, just let me share my screen. Um, just give me 1 second. Yeah. Okay. Now, just checking that you can see my screen.
We can, Pei. Thank you so much. So if you want to go to slideshow, brilliant.
Okay, thank you. So thank you everyone for coming today. I'm going to present the case of Michael, where a genetic diagnosis made at the Garvan was able to significantly alter his treatment and change his life. So Michael first came to us when he was 52, and for his whole life, he has been a medical mystery. He had always complained of tiredness, chronic sleepiness, difficulty thinking, what he described as brain fog, and also poor libido, stomach upset, and chronic bowel symptoms with diarrhea, and also sore joints. In other words, he felt that he was almost like a car running on an empty tank for all his life. In his 50s, he was also diagnosed with a very rare cancer called Kaposi sarcoma. So this is a cancer of the cells of the blood vessels and the lymphatic vessels. And classically, we see this most commonly in the setting of HIV when someone's immune system is compromised. However, Michael is HIV negative, and no one could explain why he had Kaposis or why he had all these other symptoms. So throughout his life, spanning decades, Michael saw many doctors. These included GPs, immunologists, a respiratory physician, also cancer doctors, stomach doctors, and even a psychiatrist in his diagnostic odyssey to find an answer. However, despite seeing many doctors, he never got a unifying answer for why he's always so unwell. Eventually, one of his specialists referred him to Circa, which is mentioned previously, stands for Clinical Immunogenomics Research Consortium Australasia. So it's a bit of a tongue twister. But essentially what this organization is, it's a multidisciplinary organization of clinicians and scientists, initially based in Sydney, but subsequently expanded to the whole of Australasia. And the goal of Circa is to provide diagnosis at a genetic level for patients like Michael, and hopefully, which then will lead to better treatment. So the way Circa works is that we have a monthly meeting where clinicians and scientists meet together and we discuss cases like Michael's case. So all the patients often had seen many doctors, they have many rare exotic symptoms, but they do not have a diagnosis. Then we send the patient sample for wet laboratory research and genetic sequencing. And if we get an answer, fingers crossed, then we will be able to hopefully provide better treatment for the patient as well. So Michael was discussed at one of the monthly Circa meetings, and we subsequently sent his bloods to the Garvan Institute for further research. So this is Dr. Jin Yan Yap. She is a postdoctoral researcher at the Garvan Institute, and she did a lot of work looking at Michael's bloods. And like Sherlock Holmes, she went through his immune system with a fair tooth comb to see what was going on. And interestingly, what we found was that Michael's immune system, similar to his clinical state, was also exhausted. So just to explain what we mean by that, so this is what we call a flow cytometry diagram. So what it does is we look at the surface markers of the white cells in the human body. And so they express markers like what we call CD3, CD45, CD8, and a range of other markers that we use. And they basically get transported to a chart, and we can look at the shape of the cells, and that can tell us whether the cells are normal or abnormal. So in this case, what we can see is that compared to a normal person's white blood cells, Michael's white cells were grossly abnormal. And this is consistent with what we call an exhausted phenotype. We also sent Michael's bloods for whole genome sequencing, where we sequenced 3 billion base pairs of DNA. And what we found was a mutation in the CTLA4 gene. So the DNA is the building block of your cell, and your cell uses the DNA to basically, think of it as a recipe to construct proteins. And when you have a problem in your gene, in your DNA, such as in the gene encoding for CTLA4, you can cause abnormal proteins to be built. And in Michael's case, a mutation in the gene CTLA4 resulted in a shortened dysfunctional protein. So how does not having enough CTLA4 explain his exhausted immune system? So normally your immune system, as everyone knows, is involved in fighting infections. However, its other function is, as we learned recently, is also to prevent the development of cancer. The protein CTLA4 is a regulator of the immune system. It keeps it in check and prevents it from being overactive and hyperstimulated. In Michael's case, because his CTLA4 was not working properly, this led to a chronic activation of his immune system. So because of this, this was responsible for a lot of his symptoms, including his fatigue, his diarrhea, his sore joints. But as your immune system as it becomes hyperactive, with time, it eventually, like everything, it becomes exhausted. And when it becomes exhausted, it also loses its ability to prevent cancers, which we think was what was responsible for the development of Kaposi sarcoma in Michael. So how does this help us in his treatment? So this is a very complicated diagram, but the take-home message, but what it shows is that shows is where CTLA4 sits in the signaling of your cells and and the interaction between your immune cells and your tumor cells. And when we found out Michael's issue, or his problem rather, was in the CTLA4 protein, the question we asked ourselves was, is there something we can do along the cellular pathways where CTLA4 signals? Well, can we target one of those pathways to help Michael? And indeed we can. So one of the proteins that's involved in CTLA4 signaling is a protein called mTOR. And it just happens that there is a drug that's readily available off the shelf called everolimus, which actually targets this protein. So we decided to give everolimus as a trial to treat Michael. And after starting treatment, what we found was that it actually restored the function of his CTLA4 protein. And also we found that his immune system went back to a normal phenotype without the exhaustion. Along with that, it led to the control of his Kaposi sarcoma. Additionally, in terms of his other symptoms, his fatigue improved, his sleepiness and brain fog improved, and also his stomach upsets improved, and also his joints improved as well. And as mentioned previously, his cancer also went into remission. However, there are still numerous unknowns in Michael's case. Firstly, this condition of CTLA4 haploinsufficiency, which is the scientific name for the disease, has only been described in the last 6 years. There are also still a lot of unknowns in terms of this disease, in terms of its long-term prognosis, but we know that Michael is still at risk of developing future cancers. Therefore, he currently still needs close monitoring and review. Nevertheless, by studying Michael's condition and discovering the mutation, we were able to find other patients in Sydney in collaboration with our colleagues in other hospitals, and we were able to find other patients and thereby providing them with the same treatment. So in summary, patients with rare genetic diseases of the immune system often embark on prolonged diagnostic odysseys. These diseases not only cause infection, autoimmune problems, but also cancer. And in cases such as Michael's, collaboration between clinicians and scientists in organizations like Circa are essential in helping these patients reach a diagnosis and treatment. And we do this through cutting-edge medicine and research. I just would like to firstly acknowledge Michael for agreeing for us to do this presentation, and also our numerous clinicians and scientists at both the Garvan and other institutes across Sydney. And I also would like to thank the David Cooper Memorial Fund for funding my PhD research, and the other donors for their contributions to Circa research. Thank you.
Thank you so much, Pei. A really inspiring look into the application of precision medicine through clinical trials such as Circa, research-based clinical trials. Now, shortly, a wonderful Joseph is going to bring up the poll questions so that our audience can vote. While we give the audience a chance to look at the questions, and I will read them out for everybody's benefit shortly, perhaps you could just tell me how many Michaels are there in Australia? How many people are waiting for decades to get a diagnosis and appropriate treatment?
That is a very good question, but also a very difficult question to answer, because this area of medicine has only developed recently. We also just off the top of my head, we've got at least about 4 or 5 patients just on our list at St. Vincent's in similar situations as Michael, where with different conditions, but similar similar stories in that they had diseases for multiple decades, had seen numerous doctors, and eventually a genetic diagnosis provided the answer. In terms of Circa as a whole, we've done just over 100 patients now, and who all kind of had, very unusual immunological presentations. And we've been able to provide diagnosis for 40% of them. So a lot of the patients are still undiagnosed. But I think the rate limiting factor also is that sometimes people don't recognize the clinical presentation, and often they get put into the too hard basket. So the real number is probably a lot higher than what we've seen.
And you mentioned, it really kind of resonated with me because I've heard this story a number of times. You mentioned that at one stage Michael was referred to a psychologist uh, because clearly, not having found a diagnosis, some specialists may have thought it was in his head. How often does that happen?
So I mean, again, it's really hard to know, um, like just because the prevalence of chronic fatigue in the community is really high, but only a very small number of them reach us in terms of people making making the decision to pursuing it further. The I suppose what was important in Michael's case was that the red flags that made his doctors think this is not just all in his head was the fact that he developed Kaposi sarcoma, which is a cancer that we really do not see these days outside the HIV HIV population. So, and that was the trigger that was the that was his trigger for referral to Circa. And then we initially did some screening tests, and we found those abnormalities in his immune system, which led us to do whole genome sequencing. But to answer your original question, yeah, it's really hard to know how many people like Michael are sitting out in the community.
Thank you so much, Pei. I think we've got some results coming in from the poll questions, so stand by. Alrighty. Thanks everyone for voting. So the first question, the first successful question is, from your experience, are there any clues in DNA that suggest genetic dysregulation of the immune system before diagnosis?
Okay, so so generally the DNA analysis part that comes later, but there are a number of clinical clues that tells us that there's a genetic dysregulation. So the things that make us think there is a single gene responsible, what we call monogenic, a single gene responsible for the patient's presentation. This includes younger age of onset. So if you're saying you've been sick since you're young, that's a red flag for us. Or if you've got clinical presentation that spans not just one basket but multiple disease baskets. So you've got some symptoms of autoimmune disease. You've also got some unusual rare infections, and in Michael's case, you could have an unusual cancer, or if multiple organs in your bodies are just unwell. So and and also, if multiple family members are sick as well. So that kind of, raises alarm bells for us. And so, and if you're not if the classical treatment for the condition you've got doesn't work and you're resistant, that's another red flag for us as well.
That's really helpful. Thank you. Next question. What is the process for genome analysis within the Circa program?
So we so firstly, the patients are presented at our monthly meetings, and if the clinicians and scientists agree that this patient possibly has a genomic disease, we send the patient sample to KCCG, the Kinghorn Clinical Genomics Unit, for whole genome sequencing. The process of sequencing is what we call next generation sequencing, which we sequence the whole genome, and then the sequence is compared to a normal reference genome to see what the differences are. We usually get a few million differences just because of the difference in individual variations. And then we basically then go through it with a magnifying lens to try and work out whether any of these differences could be responsible for disease. So there's a significant scientific there's a significant science part to this, but also towards the end, as you're looking at the different genes, it's also an art component to this as well, in terms of deciding what's important and what's not in the genomic information.
Hence, why it's so important to have clinician scientists like you, Pei, I would assume.
Yeah, so thank you. Yeah, so having that clinical background certainly helps, because you can, I think, filter out what's important and what's not. Just, yeah, if you know a bit about the diseases from a clinical perspective.
Fantastic, thank you. And I'm really happy with the third selected question, because it's one that I've been wanting to ask all session, which is, why is understanding the immune system important for treating cancer? There seems to be a clear connection here, but
Yep. So the immune system, its function is not only to fight infections, but what it also does is it monitors your body for abnormal cells. And when the cells become abnormal, for example, they lose surface marker, or they express an abnormal surface marker, your immune system actually recognizes this as foreign. And then it, similar to how it recognizes, bacteria, viruses as foreign, and then it goes off and kills this foreign cell. When you develop, what happens in cancer is the cells loses its normal homeostasis mechanism, and it actually starts to behave abnormally, and it starts to express abnormal markers. And then your immune system recognizes this, and then basically it goes off and just kills it like a smart bomb. I think that's the best way to describe it, and prevents the abnormal cell, basically prevent it from expanding, and prevent it from developing into a cancer down the line. The problem is that so in Michael's case, your immune system becomes dysfunctional, it loses its ability for cancer surveillance. But also what can happen in, I guess, bread and butter oncology, cancer medicine, is that some cancers evolve mechanisms to escape the immune system as well, which is why there are also treatments now on the market where they try to upregulate the immune systems to kill cancer, which I'm sure a lot of people know, for example, in the area of melanoma, where there's been a lot of developments.
Fantastic, thank you. Well, I mean, it sounds terribly exciting. What are you most hopeful for in terms of the future?
So I think there's two things. Number one is that we are hoping to improve our clinical acumen through seeing patients such as Michael's, so that we can those pick up more patients, those patients in the community who's been through what Michael's been through, who have been dismissed by other clinicians or doctors. Uh, I guess what we're hoping for is that we can get more of these patients through our research pipeline, so that firstly, it'll let us understand the immune system better, but also provide a diagnosis and hopefully treatment for them as well. And I suppose secondly is that by doing this research, we improve our understanding of both genetics and the immune system, and and allow us to improve our diagnostic pipeline as well, so that we can reduce the number of patients who we're still not able to provide a diagnosis.
Thank you, Dr. Pei Dai. A really inspiring talk. We're so grateful and privileged to have had you here with us today and to have you at the Garvan Institute and the St Vincent's Hospital Precinct. Our sincerest thanks. I'm about to get in trouble because we have gone over a couple of minutes. My apologies to the audience. So without further ado, I will say thank you for being with us today. Thank you for your continued
Thank you.
support of the Garvan Institute of Medical Research and our partners and collaborators. We sincerely value your full and frank feedback. There will be a survey that pops up at the conclusion of this session. So for everyone in the audience, please do submit your full and frank feedback so that we can continue to improve the way that we share our science with you, particularly during these strange times of COVID-19. If you were inspired today and you're able to do so, please do consider a donation to Garvan or to anyone on the St Vincent's precinct. Please do join the mailing lists. Please do share the video that will come around of today's session with your friends and family. Never before has medical research been so important, and we're incredibly grateful to the community who makes our science possible. A very special thank you to Michael for allowing us to talk about your case today. It really brings it home how important medical research is. And we appreciate that it's not a small ask for you to allow us to share your personal circumstances with our audience. So from the bottom of our hearts, thank you so very much. We hope that your families continue to stay safe and well and COVID-19 free. We've got a little way to go, and we're really looking forward to catching up with you again if you're available on the 26th of November for our next Bite Size Science, where you will have a unique opportunity to hear about the fascinating realm of science meets art in our talk on scientific animation featuring Dr. Kate Patterson. Really hope you can join us for that. Again, apologies, I've gone a little bit over, and a special shout out to the team behind the scenes, Joseph Hewitt and Priyanka Kulkarni, who make all of this possible. And again, thank you for being here. Take care.